staff. And my name is Heather Wooden. I'm the ISD Education and Meetings Coordinator. And just so you're aware about what's going to be happening today, we're going to be going over some common questions, and then if you have any questions that you'd like to ask our panelists, what you'll need to do is, if you're on webcam, you can go ahead and just raise your hand. But if you're not on webcam, you can go ahead and use the chat box and type in, just say you have a question. And then you can go ahead and wait until you're called on and then unmute yourself. So for the majority of the presentation, please mute yourself unless you are called upon to ask a question. So just a little bit about IST. IST is a membership association that gives technical guidance to professionals working in controlled environments. It is the only organization that exclusively serves professionals responsible for the design, test, evaluation, and reliability of products and systems, the detection, measurement, and control of forms of contamination associated with and resulting from manufacturing, biomedical, and industrial operations, and the science, engineering, and technology conducted at the nanoscale, with an emphasis on the facilities that serve nanotechnology, research, and development. We also help develop, write, and publish industry-recommended practices through our working groups, as well as hold conferences and training classes. We are an ANSI-accredited standards developing organization and the Secretary of ISO TC 209, Clean Rooms and Associated Controlled Environments, and the Administrator of the ANSI-accredited US TAG, the ISO TC 209. So I'm going to go ahead and introduce everyone today, our panelists that we have. So for our panelists, first, I'm going to say hi, wave Ziva. Ziva is the president and founder of MicroRite, a California-based consulting firm providing consulting and training services to pharmaceuticals, biotechnology, medical devices, and in vitro diagnostics in the areas of quality assurance, quality control, microbiology, and validation. She has over 25 years of academic, research, clinical, and industrial experience in microbiology and quality assurance. Next, we have Anne-Marie Dixon-Heathman. Anne-Marie, you can wave. She's the owner and president of Clean Room Management Associates, a consulting firm that specializes in competitive benchmarking, training, and auditing of clean and aseptic controls and management. She has been actively engaged in the field of contamination control for over 30 years. She is an IST fellow and past president and chairs the United States Technical Advisory Group, US TAG, to ISO Technical Committee 209, and she also chairs the IST working groups 18, 26, 49, and the new Clean Room Robotics. Next, we have Morgan Poland. Morgan is a principal consultant of MicroRite. Morgan brings over 30 years experience working in clean rooms and is a subject matter expert in particle monitoring. He has been involved with various clean room projects working in monitoring of program development, particle counting design, and product management for clean room related products and systems, addressing monitoring and control solutions in a wide variety of clean industries such as pharmaceutical, medical device, semiconductor, data storage, aerospace, defense, automotive, optical, and food and beverage manufacturing. And lastly, Bill Shea. Bill, you can wave. He is a senior engineer, particle technology expert at MicroRite. Bill possesses extensive engineering experience in advancing of particle counting technology through innovative product design and product management, resulting in full product life cycles. He has broad experience in troubleshooting particle counting generation devices as well as configurations, including particle monitoring systems and smoke generating devices. And just for anyone who is logged in since I've been talking, just to let you know, you may turn on your webcam if you wish. If you do have a question, please go ahead and either wave if you have a question or type it into the chat box, and then I will call on you and then you can unmute yourself. Please make sure that you're muted the entire time until you are called upon. So Morgan, you can go ahead and take it away.
Excellent. Thank you, everybody, for joining, and thank you again to our panelists. The idea came up in order to have an open-ended question and answer session with people, I think, who have a lot of experience working in a variety of clean rooms all over the world. I've known Anne-Marie since I joined IST back in 1996, and she's been a mentor to me and has a lot of experience working with her on the TAG, creating standards for the US and representing US interests in clean rooms all over the world. Z. Abraham is one of the most dynamic microbiologists that I've worked with in all industries related to biocontamination control. Myself, I've worked in a lot of different clean rooms in all industries. And Bill Shade, I asked to be here because Bill is an expert and designer of a lot of different types of equipment, particle counters, activators, samplers, in situ particle counting, liquid particle counting in all industries. And these are people that I've known for quite a bit of time. And again, one of the things we get in terms of working in clean rooms, we get a lot of questions. And I think IST is one of the premier organizations really in the world in the field of contamination control. And we thank you all for coming, and we want to open this up and start answering some questions. So do we have any questions starting out?
We did receive an email for a question. Let me see if he's online here. Just give me 1 second.
While you're doing that, I'll start with an opening.
I was going to say, is it Sassy? Sassy, are you online?
Yes, I am.
Sorry if it's... Okay, you can go ahead and ask the question that you emailed to us earlier.
Sure. I was wondering what might be, from your experience, what might be an industry practice. So, you know, we doing lab testing for, say, medical devices, and a lot of times we have to turn the room over pretty quickly after, say, a renovation of a clean room, for example, or a repair of an LFH or BSCs. So my question is, what might be an acceptable parameters to use to release the, say, the LFH for use while we're waiting on the remaining viable results? So, for example, can we use the MVP? Is it justifiable? What are other people doing in the industry?
Anne, go ahead, please.
Normally, the room, you're asking if the room can be run at risk. And
correct. And if we are doing that, what might be an acceptable means
that is
to
a very difficult question. I mean, of course, as a manufacturer, you're free to do what you want to do. The real issue is going to be the product that's released. And so if you only have part of the data, then nothing can be released. You can always go ahead and run at risk on the assumption everything's going to be perfect. If it isn't, though, then none of the results are going to be able to be used. So it becomes a significant issue. We have a similar situation in pharmaceutical. If you were to do a media fill and then decide you were going to run a process without having the media fill results, again, you can always do that. But if the results fail, everything you made goes in the trash can. So it's a big risk to you from a financial situation. I would assess it on that perspective and say, am I prepared to throw everything away should the results from that come in wrong? Did that answer your question, Sasha?
Yes, so it sounds like we should do risk management and risk assessment internally and come up with a certain answer on that.
Right, I would not do a risk assessment. I would do a financial assessment.
Got it.
Right, because the risk you don't know on the answer, but the financial assessment, I think management's got to be prepared to make it a real hard financial decision. And that's huge, depending upon how many tests you're running.
Thank you.
You're welcome.
Go ahead, Morgan.
Okay, so here's a common question that comes up. What is the frequency of cleanroom classification testing? This is a question I was asked just the other day. And anybody want to answer that, or I can go ahead and answer that?
You go ahead, Morgan.
Okay, so the frequency of your cleanroom classification is per ISO 14604 Part 1, the second edition 2015. Every cleanroom needs to go under cleanroom classification testing on a yearly basis. The caveat to that is if you're making sterile medicinal products, a sterile facility needs to be reclassified twice per year. And the extent of the reclassification is going to be dependent upon the industry you're serving and what is expected. So again, if you're making medical products, specifically sterile medical products, you're expected by the FDA as well as the European Union and any PICS member country, any country that shares the same GMP as the EU, is expected that you would be doing your grade A and grade B every 6 months.
And I just want to reiterate for everyone, if you have a follow-up question to something or you have a question, please go ahead and put it in the chat box. It's the little chat bubble on your right-hand side. Oh, we have a question. Go ahead, you can ask it.
Yeah, thank you. Morgan, you said every 6 months as per the EU GMP. I think the EU GMP 2020 says that we follow the annual thing, and that too subject to quality risk management. If you're doing continuous monitoring, you can even extend that year. Not that I agree with it. I'm a hands-on chap. I'm a filter guy. Every 6 months is right for me. But even the 14644 Part 3 2019 says annually, even for class 100, class ISO class 5. Sorry, I'm an old man. I talk class 100. Yeah. Yes, ma'am.
It's always subject to regulations. And if you're in a regulated industry, then that takes the first priority. If you're in a non-regulated industry, you are correct in saying that there are some exceptions when you do constant continuous monitoring. There are many semiconductor firms that do not do a formal certification. They simply do continuous monitoring with numerous amounts of locations. And for them, if they're not interested in following 14644-1, if they're not regulated, and if they certainly don't need 14644-1 as part of their ISO 9000 certification, then it would probably not be needed.
And be clear, the FDA, just to extend upon what Ann's saying, is the FDA states in their sterile aseptic guidance in 2004, which is still a valid document, they expect you to do it every 6 months, including GMP Annex 1. Now, ISO 14644, Parts 1 and Parts 2, the second and first edition gives you the caveat of being able to extend your as long as you have your classification testing, as long as you have a monitoring system, monitoring particles and other parameters. A parameter would be differential pressure. However, there's no way an inspector, a pharmaceutical inspector, would accept that you're not doing that testing because you haven't done filter integrity testing and those things. So that's where the example of that is, and sometimes it's confusing, is the ISO cleanroom standards are written for all industries, and then the GMP regulators have additional criteria or expectations that they try to put on top of that. And it's important to understand, even though ISO may give you that pathway, the regulators have their own expectations, and by that, expectations essentially mean requirements, unless you can prove scientifically you have a better methodology. Did you want
I get that, like the US said, sorry. Yeah.
No, yes, go ahead, Sant. Go ahead, Sant.
No, I said the US FDA says, in fact, even if you follow ISO 14644 Part 1 and 2, there's no guarantee that we're going to approve it. You can meet those requirements and still fail the FDA. Anyway, yes, ma'am, please. Just confirming what you're saying. Yes.
In the latest draft of Annex 1 2020, there's a reference to Annex 15 that specifically talks about the validation and the revalidation requirements. And I think everybody should look into Annex 15 and then compare it with the ISO and the other expectations and come up with a plan as to how, and a justification how, from a risk perspective, how often they are going to certify or qualify a room.
Okay, I mean, I vote for 6 months, so thank you.
No, Annex 15 is a good document to
Okay,
read.
I'll have a look at that, ma'am.
And
I'm
then,
not aware of that yet.
yes.
I'll have a look at it. Thank you.
All right, we do have another question that was submitted. Ricardo, you can go ahead and unmute yourself and ask it. Go ahead. I can't hear you. Oh, it's the head. If you want me, I can. Yeah. So he did ask a question about CER modification. When adding equipment to a CER, what is a sensible approach to limit the area that we should monitor for particle count changes? Instead of monitoring the whole room, perhaps limit the approach to the equipment around the new equipment. Morgan, who would you like to have answer that?
I can start. So I don't know what the acronym is of CER. I'm not sure what that means, but
hello, can you hear me now?
yes.
Control environment room.
Yes.
Oh.
So
Okay,
let me perhaps elaborate a bit on that request. And thank you, Heather. I was fighting with my mic. So the idea behind this is, well, you know, we have large rooms, and we're talking about 200,000 square feet. So when we're adding new equipment, we know we are going to change the room performance, and we have to monitor. Now the question boils down to, I won't be adding all the equipment at once. Sometimes we're adding half a manufacturing line, and a couple of weeks after we'll be adding another one. And trying to monitor the whole room every time we do a change that's small, it's get costly really fast. So the question is, what would be the right approach to limit? Well, I got my particle test location set up according to the ISO standard. How should I pick, or what's the right approach to say, well, you know what, I'm just going to limit to these points around it, and this should give me enough evidence, and I can say or prove that really I don't have to monitor the other side of the room, because that's kind of the gist. Thank you.
Yeah, go ahead.
I've done it a few times where what we've done is figure out what the impact of that equipment is on the room. And so if all your equipment is the same, then what you could do is model one, and you would know what that distance is. And what you need to do is look at, move it out start at 3 feet from the equipment. And then 3 feet, that's really where you should see some impact. And then go in a foot to 2 feet. And if you get a higher number, then you monitor there. If it's 3 feet, it's still high, then try 4 feet. Generally, it's between a 2 foot, 4 foot zone that the equipment will impact with no one moving by it, and that's the key. But you want to look at the moving parts of the machine. And in some cases, if you've got a full ceiling, it may not be impacting very much of the area. This random flow could impact more. But generally, pick a zone around the equipment and then use that. And if it's a similar piece of equipment, then I kind of standardize. So I'm a 4 foot window around this piece of equipment. Or 3 foot.
I would like. So I didn't know what industry you have. What industry are you in particularly?
Oh, we are on the medical device industry. We have classrooms with mixed airflow.
Okay. Yeah, so the thing I would add, and I've been in a lot of facilities, started in the semiconductor industry, where we have these really large clean rooms, like probably larger than you have, and we bring in equipment all the time. In fact, when a wafer fab starts up, they start running silicon often before the entire room is built. They kind of they'll build the they'll put in some of the larger equipment like ion implanters and steppers and things, and build the room around it and try and get things started up. So one thing I would suggest is to understand your airflows, because what I've seen in experience in different industries is when we bring in new equipment, we make modifications. Sometimes the process of bringing it in, maybe we have a cutout in a wall, we bring in a big piece of equipment, that can change the airflow significantly in the room. One case history I have is from the semiconductor industry with bringing in a new piece of equipment. The process caused them to block off air returns, and that screwed up the entire airflow in a large unidirectional clean room. So you have to kind of, I believe you should step back and really evaluate the risk and take in consideration, as Ann said, the particle counts is an excellent way to monitor it. But before you even get to that point, understand what's going on with your airflows, because if that new equipment has made some modification to the room in terms, sometimes that modification simply be you have a big piece of machinery that you brought in, and that machinery, blocks a lot of airflow. That's a possibility. Or, you might be putting this on the perimeter walls, blocking off air returns. Blocked air returns can significantly hamper or choke down the clean air, creating eddy currents and the possibility of contamination moving around. So, yeah, I would something I'd evaluate. The particle counts, I think, would be really good. And I don't know if we want to talk about microbial monitoring. Ziva?
So yes, so I have experienced a lot of times medical device CMOs, right? They have the scenario you are talking about. They have different sponsors. They bring in different equipment, and now they say, okay, we had a set environmental monitoring program, a set way of monitoring, and now the equipment, it is larger or smaller, more robotics or more cleaning. How do I evaluate? What Ann said and what Morgan said, a combination of both those things are very important. You might be blocking, and I have seen a lot, and I can't name anything, but a lot of such medical device CMOs where the sponsors are changing all the time. The equipment is changing all the time. And your challenge is, I'm not going to re-evaluate the whole facility. And I think a combination of what Ann said, look at what your equipment, and what Ann said is actually going to give you an idea is really how is the particulate count changing in the vicinity of my equipment, and that can address the airflows. Am I creating dead space? Am I creating turbulence because the equipment movement and all that? I will see that in particle, but evaluating airflows also gives you a very good idea if I'm really blocking the clearing of air. The clearing of air into, trust me, I've visited enough medical device CMOs where there is so much work that the air returns are blocked, right? So taking both these approaches will give you an idea as to, where I should monitor. It might, if what Ann said, 2 feet, 3 feet, 4 feet, whatever, will also give you your risk-based sampling points. You will have to change your EM, routine EM. So wherever you find more particulates, right, it's important to understand. And what Morgan said, airflow, if you have dead air, you might have particulates and microbes. And for every medical device, you have to have pre-sterilization bioburden. I hope together we addressed your question.
Thank you so much for your insights. Yes, you did.
All right,
Thank you so much. I really appreciate it.
We do have another question. Mark, you can go ahead and unmute yourself.
Thanks, Heather. So yeah, some of this is a setup just because I want to hear Ziva say Pseudomonas and Aspergillus. But this question is really related to, we've heard about microbial biofilms, particularly in environments where moisture is present. That could be, inside of equipment during manufacturing. And so the type of disinfectant validations that we do today don't really involve exposing to biofilms. Do you think that should change now that there are some models out there for creating biofilms?
So Mark, I used to teach how to do disinfectant qualification using my biofilms. I can actually send you, I have no problem. I don't teach it anymore. I taught it 10 years back where the industry was not open to testing micro. It's a total different. There are two options you have. I can send you how to create biofilms, how to test them, but you have another option to send them to biofilm engineering school, and we have a relationship in Montana. You know, that is the best place where all the guidances for biofilms are coming out from. It is an underappreciated and underutilized test, because what kills stragglers, as I say, does not kill biofilms. It's the cover, the capsule, breaking up of the capsule. So you can, Heather, if you are okay, and Ann, if you are okay, he can write. You can send me an email through Heather, and I will be more than happy to share both these ways of testing biofilm.
Thanks, Ziva. Yeah, I know about Montana State, and that they have been instrumental in creating some biofilm methods using the CDC reactor, and that's actually what the EPA now has approved, that if a disinfectant or a product wants to make a biofilm claim, that they use that type of equipment. It's fairly intensive. That's why, yeah, you generally pay somebody else to do that, not try to do that yourself. Yeah.
It's very easy. Mark Warnock is the one who developed the CDC. I had a laboratory, a training laboratory, where I used to teach all this, how to use a CDC reactor. And I'm glad that we are there, but it's best. And I can tell you the contact, who to contact, Paul Sturman from Bio, and he's the industry liaison. He'll be able to help you out.
Thanks, Ziva.
Does anyone else have any questions right now, or we can move on to some more common question? Oh, Mark, go ahead.
I'll just keep them coming, but no, I'm curious to hear what y'all want to talk about. But this one's related to obviously, we, again, being a textile provider, so we hear a lot about the debate for various reasons, quality as well as sustainability of re-laundered textiles, be they the cleaning mops, the wipes, even gowns and other PPE, and folks for various reasons wanting to re-launder those or reprocess those versus just throw them away after single use. So I'm just kind of curious about the panel's opinions on, you know, is it possible to re-launder these products and not reintroduce bioburden or linting, you know, shedding fibers back into these controlled spaces?
Go ahead, Ann.
I don't think we have a problem, but again, it depends on who's doing it and what the specifications are and how well the process is validated. And that's a big set of words I just said in a very fast sentence. But if you've got a controlled process and you've got the right kind of water, the right kind of solutions, you have the cleanability of the equipment, and you've proven that. You actually sample the rinse water, and you control, of course, how things are going in and how they're being packaged coming out. There's no reason it couldn't be, but you also have a life and a usage range for all these products, and pulling things out of their life cycle soon enough so that you don't get a crossover contamination from fibers and heavy particles. I think that's a critical component to this. And many people are not. They're not barcoding items, and even if they are, they're not controlling the cycle time. They're also not inspecting. You know, it's an easy thing to say, I reprocess a mop. And you say, well, yeah, that works fine. Well, mop A maybe was used on terrazza. Mop B was used on stone hard finish 2, but mop C was used on stone hard finish 3, and they're very different. And certainly using it on 3 of a finish for stone hard is going to shorten the life cycle logarithmically from number 2. And so I think there's a lot of awareness that the industry doesn't have today with the actual use of the product and the testing that's required and the validation by the vendor.
So true.
Thank you.
Welcome.
We do have another question from Ricardo. Ricardo, you can go ahead and unmute yourself again. I can't hear you.
No, Heather, he typed it. Why don't you read it?
Can you hear me, guys?
Yes.
Yeah.
Oh, sorry. Still battling with the computers. So on the installed filter leakage test, again, we have some rooms that really an upstream aerosol challenge is not at least easily feasible, either because the room's already fully loaded and we really don't want to be lifting the ceiling tiles to be able to inject, or there was no access port readily available. You know, you name it, there's a lot of things that make it harder. What are alternate options for doing that? I was trying to go over the IEST recommended practice 34, and trying to look at as well on the ISO, the -3 for the test methods. I believe there's a light scattering air particle counter method, but I really would like your input to make sure I'm not misunderstanding.
Yeah, so here's the thing. You're a medical device manufacturer, correct? So in your question is the alternate method, you have to challenge, you have to do a filter integrity test, right? The FDA requires it. And you have to do a filter integrity test with an upstream aerosol. There's no way you can avoid it, right? So whether you could use an alternate method, you could use an airborne particle counter and a different challenge aerosol, but regardless, you still have to have an upstream aerosol challenge to test your filters. So that's going to be a challenge you're going to have to face, because that, in case if you have a plenum-based system, we have individual filters. You have, I'm assuming with the plenum-based system, you don't have FFUs, you have plenum-based filtration, right? So then you have to, so that's, I'm sorry to say that, that's a really outdated design. I'm sure you understand that, because you can't challenge. The beauty of individual FF fan filter modules is I can test each individual HEPA filter and generate the aerosol at that fan filter unit. When you have a plenum-based design, you got to generate so many, such a large volume of aerosol challenge that the filters you're testing, so you're going to test all these filters in one plenum. The process of doing the testing decreases the life of the filter, because you're draining that test aerosol for a very, very long time. But that's the weakness of your system. And the only thing I can tell you is, when you have a chance to modify your facility down the road, take that into consideration and get away from doing, you know, those large plenum systems, because you're required to, because you're a medical device, to do this testing on an annual basis. And I don't know if Anne, you wanted to share something related to that as well.
You know, my advice to you would be to get somebody in there and see where you can install some ports. That would be my advice, because they do need to be there, and you can install the ports after you've completed the clean room. I'm not saying it's easy to do, but you could have someone come in and take a look at where the ports can be installed, how it can be done so it meets your requirements. And so, yeah, it's a requirement.
There's actually a couple companies that sell ports to modify your system. I'm not familiar. I've seen them. I don't know the company. But
yes, I have also, and I've seen people who have done them. And again, you have certain certification firms that have the capability of going in and reinstalling ports for you at this stage. And so my advice would be to bring somebody in like that who knows how to fix those without damaging your filters.
I appreciate it. Yeah, if it's, you know, something we got to live by, it's good. And I appreciate the input on the access port option. That could definitely help alleviate some of that trouble. Thank you.
All right, we do have another. Yeah, we do. Patricia, you can go ahead and unmute yourself and ask a question.
Yeah, I was actually just wondering your thoughts on the need for cleaning validation studies, specifically looking for residues or residual removal for a facility where we don't have any product contact surfaces within like the equipment or the facility. And basically all of our materials that do have product contact are sterile single use. So what are your thoughts on doing any kind of cleaning validation and where those would be most beneficial considering the situation we have?
Can I answer this question?
Please.
So, Patricia, are you talking about cleaning validation or disinfectant qualification? These are two different things. Cleaning validation is for equipment where there is product contact, and disinfectant qualification is for hard surfaces in the clean room. So can you expand on that question?
Kind of both. So the first question is exactly as you're saying. So we, I guess to give a little backstory, we had someone come in and they were asking for our cleaning validation, and you know our defense was kind of, well, we don't have anything that's product contact, we don't have any equipment, and so we were talking about our single use materials.
Yes.
That's kind of where this question was going. But yeah, I do understand kind of more the disinfectant efficacy testing. That's, I don't necessarily have a question on that. That's one that's kind of a work in progress for us. We're just not quite there yet, but more so the cleaning validation, how you would kind of justify not using that if we don't have product contact.
If you are using single use and you have no product contact, right? You throw away whatever you use, you don't need to do cleaning validation. But when it comes to, and I will tell you about disinfectant qualification study, because according to me, it is the most overrated study, which really does not give any guarantee of contamination control, because it's very subjective. It's done in different ways, and the data can be all over the place. You know, I used to be a part of the biopog where we wanted to refute, companies get together and refute that this study should not be. And I still, since 1998, I wrote the first chapter 1072, the first draft for USP. That was the first disinfectant qualification draft. And it has become a checkbox for the regulators. I would ask people to do a bracketed approach. As Anne said, all the material, she just talked about stone hard stone tech. We put it in one box, right? No, stone hard stone tech itself is so many different, you know, the roughness, the softness, the how much the material soaks, right? Will change your recovery. So that is something I tell people, please take a bracketed approach. Don't go overboard with it. But answering your question, cleaning validation, you can easily justify if you're single use, you're never going to make another product in that container or in that bag. So, and if you have any valves or components that are product contact with different product types, that is something you have to assess risk and show that they are clean and the residue is removed, and have a specification of how much residue is allowable.
Okay, perfect. Yeah, that was the defense that I used, but I think the person that I was speaking to was so used to being in facilities where there was product contact. So I'm happy to hear you say that. And kind of where you touched on saying disinfectant efficacy is kind of a checkbox. We're gonna, you know, check that box off, but we're looking at microbial flora assessments to look at the real world, what's going on in our facility to actually see the microbial load and the effectiveness of our cleaning and disinfection.
Trend, and don't expect miracles, because I don't want to expand too much on it because there are many people waiting. But the way the AOAC testing is done for label claim, the organisms are weak organisms and we see tough organisms. So do not be surprised if you don't have kill in 10-minute contact time. So don't limit yourself to 10-minute contact time. Just do it as a study to understand how long it takes for the disinfectant to kill the organisms you choose, and develop your disinfectant program accordingly.
Perfect. Thank you.
All right, we do have another question. Hey, Zeus, you can go ahead and unmute yourself.
He says he's having microphone problems. I can read the question.
Oh, okay. Oh, I see it now. Yeah. So the question is, for grade 8 room isolator background, to determine the number of EM samples per ISO 14644-1, can I remove the area of the isolator from the entire room to estimate the minimum EM samples?
Oh.
Go ahead, Emery.
Go ahead, Ann.
Okay, so we've got some confusion. So let me make sure we understand. 14644-1 is it's a classification document, and so under classification there is a requirement in Table A1, but it has nothing to do with routine EM sampling because this is only involving particulates. So let me assume for a moment, Jesus, and you can write us back, because I think the whole panel is sitting here agreeing with me. So you're talking about qualification. Okay, very good.
Yeah.
Okay, so we're back to the classification / qualification of the room itself. You know, the isolator as a footprint, because of how it's designed and the fact that it goes up into the ceiling, actually occupies its own footprint with its own air. So when you look at the 8 background or the 7 background, depending upon what you have, generally you do deduct that piece of equipment because it is its own self-contained unit. And then the number of samples to meet the Table A1 is a calculation based upon the remaining area. Did that answer your question? Since he's having microphone questions? Yes, he did. Okay.
I would like to add just one thing. Now, the Table A1 in ISO 14604 Part 1, 2015 edition, the second edition, that's the minimum number of locations. One of the things I try to tell people, or I believe, sometimes the minimum number of locations may not, there's no reason you can't take more locations. And again, this is one of the reasons when you set up, you do your qualification, you may find that, hey, I have a lot more activity, I may want to do additional locations. Or when you subtract out area, or if your room's oddly shaped, you may find adding more locations gives you better coverage for that. But again, for the qualification aspect, you know, you need to hit that minimum or exceed it. And again, factors like risk, activity, things like that. If you have an area that you think might be dirty because of the type of operations, you may want to have more points. So just to expand on that a little bit.
One quick comment, though. I hope everybody on the line, whatever you do not certify a room with 1 sample. To take the advice of people who have been there and have waited a very long time for the particle count to achieve the minimum number. And in some cases, if the room is very clean, it could be days. So please always take 2, even though the standard allows you 1. Go ahead and take 2. A little bit of humor, right, Morgan? I said, Morgan, it's a little bit of classification humor, right?
Yeah, exactly. Yeah. I'll sit there and wait till I get a good count. I don't like those results, keep testing.
All right, we do have another question. David, you can go ahead and unmute yourself.
I do have 2 questions. One is to piggyback on the disinfectant qualification. It could get very, very expensive very quickly. So how do we limit the number of surfaces that we're testing? Example, if I'm looking at a clean room door, you have a knob, right? So I have a door, I have a knob, and the surfaces, I have a bin that I put material into, but they are in bags. So what is the limit, and then how can I justify it? Okay, I'm only going to do this is surfaces instead of 9.
But trust me, I know how expensive I just want to give you a case study here, okay? There was a company that got consent decree. They had spent $3 million on the disinfectant. Hard to believe, right? They took all the organisms, all the surfaces, all the disinfectants, all the dilutions, and sent it to a lab that was not qualified to do disinfectant qualification. You have to understand the people sitting at the lab, at the bench, are not disinfectant experts. They are not clean room experts. They are not cleaning experts, right? So they are running a protocol. One of the ways we do this, and we do this all the time, is take a risk-based approach where, where my recovery will be the roughest, right? Least recovery. So as Anne-Marie said, which is the roughest surface that we have where my recovery will be least, right? Then I have to look at, not all my disinfectants have surfactant in it, so they will not spread evenly, correct? So which are the surfaces which are smooth, because they will have surface tension. Then which are the surfaces that will soak up, right? Fast, correct? Which are the surfaces I cannot sterilize? So you have to do a sort of risk assessment and come up with a bracketing approach. You might have 10 surfaces which are smooth. Do you want to test all the 10 surfaces? Absolutely not. You take the worst case scenario. As the rough surface, you figure out what is the worst case scenario in roughness and come up. Then you look at what are critical surfaces. The closer to the open product, you want to test it. And come up with a rationale, justify it. Organisms. Do you need to test 20 organisms? No. You can take some from the USP, and I don't like the changes that have come to 1072 right now, because they've made it into like, oh, it's a mandatory. It is the, can I use the word silly? It's a silly test. So you figure out, I will take a few and do a combination of your organisms and USP organisms, and in your organisms, take the top, do the predominance, the top bugs that you are seeing.
Okay,
and don't expect miracles, please, because not all organisms will get killed at 10-minute contact time, which is a misinformation in our industry. Some will take 20 minutes, some will take 1 hour, and some won't get killed. So does that mean that you're not going to use the disinfectant? No. You have to then use this data to develop a science-based disinfection and qualification and cleaning program. Anybody wants to add anything more, Anne?
But you know, Ziva, I so many times people will test 316L and they'll test 304 stainless. And, you know, there's no doubt 316 is very easy to clean. And so if you're going to test one, you test the tougher one. And Ziva's so right. We need to categorize them. We need to group them. We need to look at our surfaces. Unless we've got millions of dollars to throw at DET studies and years for them to be done, much less being able to get the coupons, which is in itself is a feat. No, we need to think about what we're doing and why we're doing it and what we're trying to prove.
Correct.
You had another question, David?
Well, let me add one other comment. So Steris made the change, right, from vaccine 2 to 3 and pH 2 to 3. Right. So we're gonna test those, but we have never done our disinfectant qualification, and we also use IPA, so we have to test that also, right? So I think that's pretty common. So it could add up pretty quickly. That's why I wanted to throw it out there, just to see what you experience with what you have seen.
Oh, yes.
What is the expectation from the FDA?
Did you test the original under Steris before, or
no,
did
we have not
tested it at all?
We were not aware of it until a consultant told us that it's required.
So, choosing a contract lab is another challenge. There will be one contract lab which will charge you $250,000. There's another contract lab which will charge you $60,000. Got it? Is one better than the other? Not necessarily. You have to understand this is a very big money maker for the industry. So you have to understand what method they're using, how they're validating the method, and how good is the data, right? So review the data. If you, for example, if you're using Staph aureus, and you say, oh, you know what, my sporicidal agent is not killing, then the lab is doing something wrong, right? You have to understand the nuances of this test. I wish I can share my chapter, but PDA will hang me. It tells you all about what you have to be careful with when choosing the lab, what you have to be careful with choosing your surfaces, your disinfectant, your organisms, and how to review the data and how to use that data for your cleaning and disinfection.
Do you have any information you can share with us?
I can't. I will be hung in Times Square. I don't want that.
Do you have one of the suppliers that I'm looking at is Analytical Lab Group? Are you familiar with them?
We really can't.
We cannot suggest labs. That's a problem.
Right.
You have to do some background work, and Anne is right. It's not fair.
Mm-hmm.
But you have another problem, and that is that right now, getting a lab that's willing to do it is really difficult in the stages of COVID, because many of the labs do not have the people on site anymore. And there's no way you can do testing without people in the lab. This is a very high labor intensive test. It requires a lot of manpower to do this. And if you can wait until people get back in large numbers in the labs, you probably have an easier time. I know one of my customers just sent in the coupons, and they told them it would be a minimum of 9 to 12 months.
Oh, yeah.
Okay.
Yeah, so this is a bad time to try to get anybody to do anything.
For now, start planning, David.
Yeah,
so whoever audits you knows that you have the intention.
Okay.
Do you have a comment, Mark?
Yeah, I mean, I guess I might say, David, at least there's a set of disinfectants that you plan on using. Talk to the manufacturer of who they have worked with in the past. Most of the companies that manufacture disinfectants have at least had experience with the different companies that are out there that do validations. They don't necessarily have a stake in their game. Now, steers can be a little different because they might do the validations themselves in one part of the company versus another. They do. Other manufacturers may give you an objective idea of some of the labs that are very effective but not so cost restrictive. So again, you got to be careful about giving opinions, and I understand. I value the panel not getting involved in that because we all rely on these testing partners. But in the industry, there are labs that are known to be more experienced than others. Let's just put it that way.
Yes, and you have
to have the issue with the COVID, and all that's going to be slowed down for now. But there are some labs better than others in terms of their experience and ways to not break the bank on this thing.
David, I think Ziva would agree with me. I would do two things. The first thing I would do would be to issue a CAPA on DET, and that actually takes it off the plate short term while you're working on it with a timeline, and at least have a proposal written. And that at least the agency will look at that and amongst the issues we're going through today, and probably say at least that they understand you're trying. That's the first thing I would do. The second thing I would do is follow the contact time of the vendor at the moment. because at least then you're not pulling a contact time number out of the air with no data. At least you've got the vendor behind you short term. And again, that may get you through this short term problem.
Yeah, and the planning
Okay.
itself will take some time, David.
Right.
You know, the advice Anna gave you, use that, come up with a plan, right? So when you go to get a quote, you already know your matrix, what you want to test.
Yeah, and I'm working on finalizing the strategy too. And I've also got a quote from Steris. We have talked to them and they have shared all of their efficacy validation. So we try to use what they have as a guide as well.
That's all you can do.
Right. Second question that I have is on the EM sampling frequency for medical device versus combination product. So we have a ISO 7 clean room for class 3 devices. So we have a main clean room that we do medical device, and that device goes into our second clean room that becomes combination product. Currently, we're doing our EM monthly, but combination product is following a different guidance requirements. So we need to increase our frequency. What can we do to balance the two without overburden our main clean room, which has a device product?
First question, can I say one thing? How have you submitted this product as a device? If it is a combination product, as a device or as a drug? What are your submissions look like?
It's going to be for drug, and we're still
It'll be a
waiting for
drug?
the approval. Yes.
Okay, so
Well, it's combination, right? So it's pharma.
yeah, but remember, when it's a combination product, sometimes people, companies will do their submissions as a device. It's a faster track. But if it is a drug, then you have to, the whole scenario changes when it comes to environmental monitoring.
Right. So because of drug, so we have to increase the frequency of sampling. But on the device side, again, it feeds into the combination product. So the device has a separate clean room. Do we have to increase our EM frequency on the device side because of the combination product that this device is feeding into?
So the device side gets sterilized before you fill in the drug, correct?
No, both products are terminally sterilized.
Okay, okay. It's a totally different scenario then. It's a totally different. So you do pre-sterilization bioburden, correct? And you do
Yes.
microbial testing on the drug product.
Yes, finished device.
Correct? Yes. So all your frequency, if you have your pre-sterilization bioburden trend and your drug product bioburden trend, and that brings it together, right? And you combine it and you say, okay, this is what my final drug device combination bioburden is. Where does the frequency of my monitoring and the site selection meet, will let me know if there is an increase in bioburden or not. And that should be your starting point.
Okay.
You always start with the product, you know, the environmental monitoring. We all say risk-based, 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 but that is the risk. You don't want your bioburden to increase. What are you doing now? What is your bioburden trend looking like? Do you need to increase or do you need to keep the same? Am I making sense, panel?
Yes.
The problem with the pharma side is that the requirement is you have to have more frequent sampling. of EM, including up to daily monitoring when there is, let's say, a failing process.
Oh, of course. That's a different story.
Okay.
We're
All right.
yeah,
Yeah,
we're
we have
running
time
out
for
of
one more
time.
question.
Any additional questions? If you mail the questions to Heather, we can respond to the question. Anyone has additional questions after this, you mail them to Heather. We'll circle them and be happy to answer them via email. That's okay for the rest of the panelists.
Yeah, so Patricia and Ricardo, I know you sent questions, so we will, I'll copy them and then send an email to the panelists and copy you in on that email, and then they can respond to the question for you. But yeah, thank you so much, everyone, to our panelists today. It went really well. I think we should definitely hold another one of these Q&A. But thank you everyone who attended and participated. And like we said, if you think of any questions or something like that, you can go ahead and email me directly. I'm the one that sent you the link to this meeting today, and we can get you in contact with the panelists. But everyone have a good rest of your day and enjoy your week. Thank you.
Thank you, everybody.
Thank you, everybody.
Thank you.
Thanks, Bill. Thanks, Dan. Thanks, Eva. Thank you, Heather.
You're welcome. I think it was good.
Yes, I enjoyed it.